Greetings and a hearty welcome from our Customer Experience Center in Vienna. I'm Antoine Pacheco, the Global Platform Manager for Clinical Flow Cytometry. I'm currently in our state-of-the-art flow cytometry lab, which spans 233 square meters and is dedicated to live events, where customers can explore our workflow solutions in gene therapy, extracellular vesicles, and clinical applications. Depending on the lab size, assay menus, and objectives, we offer solutions to enhance efficiency for clinical flow cytometry, with application-specific technological advancements. Here, you can see our Atrios CL load and go flow cytometer designed for automated lymphocyte subsets and stem cell analysis. Next, we have the CellMec SPS instrument, which fully automates complex sample preparation procedures, including RBC lysing, cell staining, and washing. And now, let's introduce the star of today's session, the DxFLEX flow cytometer. It's finally arriving in the United States, completing our global presence. One of the first things you'll notice is its compact design, which saves valuable lab space and ensures easy of use even in smaller labs. With a weight of 36 kilograms, it's easy relocatable. Let's take a closer look at the instrument. So the DxFLEX operates without a large, noisy compressor card. As you can see, there is nothing in the bottom, thanks to the built-in peristaltic pump that efficiently delivers cells into the system. No need to check a separate compressor or switch additional devices on or off. Just press a button and the system will start. The sheaths and waste tanks on the side are easily replaceable. The DxFLEX allows you to add more columns to your panels without adding complexity. It simplifies high complexity testing. And I will demonstrate how it works now. Equipped with 3 lasers and up to 13 colors, it is CE marked for in vitro diagnostic use. In the US, the DxFLEX flow cytometer is intended for 10-color immunophenotyping with the ClearLLab 10C reagent system. Fluorescence channels FL11, 12, and 13, and all other uses are for research use only. Now let's have a look inside the system. So if I open here the cover, the lasers are in this box, the 3 lasers, the red, blue, and violet laser. And you see here for each laser, we have the optics here with the famous avalanche photodiode detectors. So each laser has its own detection module with small optical filters and avalanche photodiodes. You can see here on the right side of the system, this is the autoloader. It's the famous known carousel from Beckman Coulter. In the carousel, a 32-position tube, you have a triple identification with barcodes. So inside the system, you have a barcode reader, and you can read the carousel number, the position of the tube in the carousel, plus eventually the barcode on the tube. And as you can see here, we have here a single tube vortexing. So it's a real vortex who takes each single tube and vortex it individually. As you know, it's really important for critical applications to maintain the homogeneity of the sample. In the B-cell webinar, we demonstrated how fast the startup of the DxFLEX is and how easy the QC and standardization process are. In the T-cell webinar, we focus on the digital data transmission. And today, we'll highlight more unique features of the DxFLEX, and we'll create an experiment with a panel of multiple tubes using the M1 and M2 tubes from the ClearLLab 10C system. Before starting with ClearLLab 10C myeloid tubes, I would like to demonstrate the power of the avalanche photodiodes. how the DxFLEX simplifies flow cytometry by gain-independent compensation. The gains of the APD detectors used in a DxFLEX flow cytometer can be calibrated for a linear response, leading to the predictability of the signal. So what I have done, I have prepared an 8-color sample, and I will show you now what the power of the DxFLEX. So let's start first by this tube 8 color. So I will select the carousel first, then select the tube with in position 1, put it in position 1, and start the acquisition by selecting the run. Okay. I can close this window here, and let's see. So the protocol is already set up. It's an 8-color panel. The system will start the acquisition here. You will see the different populations. So acquisition is starting. You see here lymphocytes, monocytes, neutrophils. And you see some graphics for compensations here. Okay. So the beauty of that, of the avalanche photodiodes, let me first open the compensation window. You see here the values actually of the compensations who have been set. Now, if I click on the graphic, this one is the APC Alexa 700 versus APC. I click on the gain settings, and if I change the compensation here, the gains, sorry, here, you see that the system will recorrect automatically the compensation values based on the new gains. I can do it multiple times. So now, the good feature of the system is if you need to adjust the gains for any reason, you can do it easily because the system is able to readjust the compensations based on the intensity you have adjusted. So this is really the power of the avalanche photodiodes. So let's stop here. So remember, for ClearLLab 10C application, specimen by specimen compensation settings is not recommended. Once the system is standardized and verified, any deviation from expected pattern should be investigated to determine the appropriate remediation. This is really a very nice, a unique feature. So let's now go back to the ClearLLab 10C myeloid tubes. Go now for an experiment with multiple tubes. I will show you with the M1 and M2 tubes, the two myeloid tubes. I will run the ClearLLab control cells, the abnormal one, with both tubes, the myeloid 1 and myeloid 2. So I have already prepared both tubes based on the instruction for use. So I have the abnormal M1 and M2. I will put it in the carousel in position 1 and 2. I have already prepared here a template, an experiment, which is called the myeloid tube, where you can see here, I have two tubes in this experiment. And if you look for the report, I integrate in one unique report both results from the tube M1 and M2. So I have the graphics of all the single color, all the colors from the antibodies, plus a statistic box where I combine the lymphocytes from the tube 1, tube 2, and also the monocytes and the neutrophils. So let's see now how it shows. So I select the carousel number 1. Here I select my panel, all the tubes, so both tubes. I put them in position 1 and 2. Press OK. I'm ready. So now just need to press initialize. Takes 2 minutes and auto record. So the system will acquire first the tube M1 from these abnormal control cells. The same strategy from the templates available in the Beckman Coulter website, where you see here forward scatter doublets discrimination with the area and the height of the peak. You have also the forward scatter, side scatter to eliminate debris. And then you have the cartography of the cells again with lymphocytes, monocytes, neutrophils, plus the immature cells here with a dim CD45 expression. I will put more cells in their position. So you can see here we have normally abnormal cells. So if we check here the CD34, as you can see, we have abnormal populations. Okay, and at the end, so I will not acquire too many cells. It's not needed right now, just to show a combined report for two tubes. I will stop it to go to the next tube. Same getting strategy. Again, you see here the different white blood cells populations. And again, normally, if we look for the 117, we see an abnormal population here. Same thing for the CD34. So we see these abnormal cells. So again, I will not acquire too much cells at the end. It's not needed, but if I press stop now, if you go to the report, the system, you will see, will combine. On the top here, I put the graphics from the tube 1. On the second line, from the tube 2, where you see FIT-C, PECD, and PC5.5 with markers depending on the tube. Same thing for the other colors here, where you see the different markers. And here you see the statistics combined. So 31% on the lymphocytes from the tube M1, 32% for the tube M2. And same thing for the monocytes, 13%, 15%, and 47% and 50%. So you see here, it's easy to combine in the same acquisition a report for both tubes or multiple tubes. From Beckman Coulter website, you can download ClearLab 10C templates for QC control cells and data analysis and reports for Kaluza C. Kaluza C is available under a single license or network licenses and can be installed on offline computers dedicated to flow cytometry data analysis. It's part of the validated ClearLab 10C system workflow.
So let's start Kaluza C. In the QC report activity, we'll see here, I have already some data. So I will go to my folder and we have already acquired control cells. So let's go for the abnormal. So it's the abnormal MT2 tube. So let's put a second file here. Okay, so what about the data? Just go to this report sheet and normally you see here all the expected values, and if we are out of range, normally the lower and upper limit values will appear in red. So for the quality control, I replay only the M2 tube, and the abnormal data. So let's go now to data analysis. So remember, to simplify all the process of the ClearLab 10C, we provide all the templates for the QC, but also for the patients, for the data. So let's go now to analysis activity and open the protocol. So the protocol is we can take the M1 tube. The last version is the version 7 right now, available in the Beckman Coulter website. And let's replay data. So remember, we provide also a casebook with multiple cases. And in this casebook, we provide also the raw data. So first, I will use probably the M1 tube here, and we provide normal samples, so bone marrow, lymph nodes, and well blood. So here I will use the bone marrow ones. So we see the data again, verify that the gating is still approximately good, so no problem here. One of the function in Kaluza is if you click right and you see here the resolution, if you have too many events, you can still increase the resolution to reduce a little bit the display and to better see the different cluster of cells. So this is the case here. So let's go back to this region here. So no problem for the gating. Seems to be nice. So it's a normal bone marrow. And we see here the expression of the different type of cells with the CD7 for some cells. We have the 64 for the monocytes. Normally there is no CD34 cells. And what's nice here, I can show you also a good option on the system. So for example, let me just create a new region. So I will create here just a region. So I just click right, you see this radial menu. I select, why not, just the rectangular region. I will select these populations here, okay? So all the cells who are CD34 positive. So if I click right and I click here, I can change the color. This is the first point, but I can also ask to show as rare event. So remember one thing is the DxFLEX can acquire up to 24 million events per data file. So it means that you can really collect a high number of cells. And if you are visualizing low density of cells, it's easy in Kaluza just by selecting this option. So just click right, go to the display. And here, if you checkbox the show as rare events, the system will change the resolution of these specific dot cells and will highlight them in all the graphics. So you can see here where these cells are appearing. I'm not really surprised because it's a CD34 positive, so you see they are dim in CD45, so probably it's hematopoietic stem cells. So now the question is it normal or abnormal cells? I believe that these ones are normal because we are in a normal bone marrow. So you see very well where these cells are fitting. So you can still change the resolution of that by just clicking right, go back here, and you see that you have here a smoothing factor. So you can, if you have really a poor number of cells, you can increase the size of the dots. So if you are in a minimum, you can still see them very well. If you have few cells, you can still increase this resolution. So it's a nice tool to visualize quickly where the cells fits in all the other graphics. So CD34, very nice, and you see also where they are fitting for the other markers. So this is a very nice option. So this one is the template for the data analysis provided in the Beckman Coulter website. If you go to the report sheet, you can still see the different sheets dedicated for the reporting. You can also, remember, through Kaluza, send all the data to the alias if it's needed. Here, you see the different subpopulations of the leukocytes, including the granulocytic populations, lymphocytes, monocytes, and the dim expression of CD45 cells. You see here, after that, some other populations, mainly based on the CD34 expression, also CD64 and CD7. And after that, we go more in details. For example, in the lymphocyte subpopulations, we are looking for the sub-expression of the CD11b, 7, 34, 10, etc., etc. So we have multiple sheets here dedicated for different lineage. So here we are in the sheet for the monocytic lineages, where we see the co-expression of the markers for the monocytes. We have also for the granulocytes here, the subpopulations of granulocytes. We have also the CD45 DIM, so where normally fits all these immature cells. So here, these are the reports. So let's now analyze another patient. We have here in the case book, we provide the case number 7. Let's just replay one data, is the first tube, the M1 tube. So I will put it down here. It will open without any protocol, and I will drag this protocol down to the sample. So just move and drag it down, and I change the protocol from the first tube to the second tube. So I can switch also, this one is nice. Here it's a density plot. So if I click right on this plot, I can go here and reselect, for example, to use the gating color. So it's nice because you remember I put in green the monocytes, in blue the granulocytes, in red the lymphocytes, and here usually in purple the immature cells. So let's put out this display of rare events. Okay, and probably let me erase my region. Okay, so what we see here, we see a high proportion of monocytes, around 43%. So we have much more monocytes than expected. And we see also an immature population, a population with CD45 DIM. And if we look for this population is CD34, has expression on HLA-DR, is also DIM in CD10, so it's abnormal expression also on these populations. It's also CD13 a little bit positive. Okay, so here this sample is identified as AML, so acute myelomonocytic leukemia. So we have the second tube, but we have no time for that. So it's just to show you the quality of the data. And if you are looking for, again, population who is not expressed too much, for example, I will take this one here. You can, again, select it, click right, choose the color to see it properly, and just display as a rare event show. So you see where these cells are fitting inside the space of the different markers and clusters.
Thank you very much for attending this webinar. It's part of 3 sessions of our Leukemia and Lymphoma webinar series. We hope you enjoyed the program today and you are listening in again.